Thanks everyone for joining us today. We'll get started in just a couple minutes. All right, thank you all for joining us for today's quantitative seminar. My name is Cheryl Barnes, she, her, and I'm a postdoc with the University of Washington and the Alaska Fisheries Science Center. I'd like to start today's seminar with a land acknowledgement, while noting that land acknowledgement is in no way sufficient to repair centuries of displacement and dispossession of Indigenous peoples who to this day still suffer from the legacy of colonization. It is, however, important to recognize the longstanding history that brought the University of Washington and many of us to this place, and to recognize those who are the original stewards of these lands. We all are sitting, standing, or connecting to others on the land of the Coast Salish peoples, land that touches the shared waters of all tribes and bands of the Suquamish, Tulalip, and Muckleshoot nations. And I have the honor of introducing our speaker today, Mike Nishizaki, who will be talking to us about more than just warming, the physiological responses of mussels to variable thermal environments. And Mike earned both a bachelor's and master's from the University of Northern British Columbia and his PhD from the University of Washington, where he worked with Emily Carrington studying the thermal biology of barnacles. Mike went on to do a postdoc at the University of Guelph, modeling fluid dynamics and zebra mussel feeding ecology. He then served as a visiting professor from Bowdoin and Williams colleges and is now an assistant professor at Carleton College in Minnesota. And today, Mike will be stopping periodically for questions, so feel free to type your questions into the chat and we'll get those over to him. And Mike, I just want to thank you so much for giving us our time today, and you can go ahead and take it from here.
Great, thank you very much for that. All right, let me share my screen here. Okay, can everyone see that in full screen? Hopefully. All right, yeah, thanks very much. Like the intro said, I'm at Carleton College, and it's a primarily undergraduate institution. So the work that I'm going to talk about today is going to be driven largely by my students at Carleton. They're all undergrads, raring to go. So the place to start is that nature is variable. Conditions out in the environment are variable in nature. Mean conditions are certainly changing in this era of climate change, but so too are the patterns of variability around those means. And so here's a little video of my site at Narragansett Bay in Rhode Island. And so I work on mussels. I'm an intertidal biologist. And so here you can see the rock, and then you can see the black mussels clinging on for dear life at the top of the rock. And so these intertidal systems are by their very nature, because they're partly in the air, partly in the water, very dynamic, and the environmental conditions are changing. So if you look at a place like this mussel bed in Narragansett Bay, whether you are in the subtidal region, the intertidal region, or in these tide pools where these mussels often live, you're seeing pretty dynamic change in a lot of the environmental conditions. Changes in temperature, changes in pH, changes in oxygen. All of these things are challenges that any organism living in the intertidal has to deal with. Organisms like mussels that can't move easily are even more at the mercy of these environmental changes. And so one point I wanted to make here is, if you look in the top right-hand panel there, you've got surface temperatures, water temperatures from a nearby buoy in Narragansett Bay. And so you see there's seasonal variation up and down, going from just above freezing to 20 degrees Celsius. But if you look at the bottom panel there, these are temperatures that I measured in the mussel bed. And this is a shorter time series. It's only in 1 year here. But you see that, yes, there's the seasonal trend, but there's a whole lot more variation on more of a daily cycle. And so some of the temperatures there are rising to nearly 30 degrees Celsius. And so some of that's water temperature, some of that is air temperature when they're exposed to air and warming on the rocks. So certainly there are changes in mean temperature, but there's a lot of dynamic change, even on a daily cycle. So my talk today, the experiments that I'm going to be chatting about, all center around marine mussels. And so I've got some examples from the East Coast, Rhode Island, where I do some work, and I've got some examples from the West Coast at sites up in Friday Harbor. So there are two species. East Coast is Mytilus edulis, West Coast is Mytilus trossuus. They're closely related, but they are different species that I'll be talking about. So mussels are important for a number of reasons. From more applied aspect, they're food. Many of you probably, this is how you know mussels, through eating them at restaurants. And so the supply of mussels in the US comes both from aquaculture and wild capture. Here's some stats that I pulled off of the FishStat website. And so what you see here is that there's both aquaculture and wild capture. Most of the aquaculture actions taking place in New England and on the West Coast. And so places like Taylor Shellfish and Penn Cove here in Washington State are certainly players in that. And so here we have some examples. You got Penn Cove, which is a local place. This crazy mussel farm. I think this is in Europe, where they actually pull the entire mussel population out and pull it out onto a boat. So advantages of having slow sloping beaches. And then there has been talk both internationally and some test cases domestically about offshore mussel farms being deployed a little farther away from the coast. So from an aquaculture fisheries point of view, mussels are definitely of interest. Some people are looking at mussels not just for the food they can provide, but for some of the effects they can have on water quality. So mussels are suspension feeders, right? They're pulling in water through their inhalant siphon, sieving out some of the particulates, and then pushing the feces out their exhalant siphon. This removal of material from the water is an interesting biological process. So whether it's through mussel farms or just outplanting mussels into sites that are of interest, there's a potential for mussels to remove some of the nutrients from the water to help mitigate the effects of eutrophication. And so people have been studying this. Here, this table is looking at the cost per kilogram of nitrogen that can be removed from mussel outplanting. And so there are differences in the cost between winter and summer, so that depends. But those numbers there in the cost are not off the charts, and this is a bit of an older study, but it suggests that this could be a way to complement some of the more familiar traditional land-based techniques for mitigating eutrophication and nutrient pollution. So mussels for food, mussels for bioextraction. When we go back though to the ecology of these coastal sites, mussels are important foundational species. So on many shores, they are the numerically dominant organism on the shore. They make a very familiar black zone that you can see at most coastal sites with hard substrate. They provide habitat for other organisms to settle in and recruit, and they provide refuge spaces for other invertebrates and for seaweeds. So beyond just the applied aspects, they're important ecological players. So I'm going to talk about these two sites, East Coast, West Coast, and there are mussels at both of these sites. I'm going to be talking about the mussels that are living in the tide pools, because the tide pools themselves undergo pretty dramatic environmental change on a daily cycle. I'm going to be focusing in on temperature as the environmental factor that changes. And what I've got down here on the graphs are temperatures that we've monitored in the tide pools of these two sites. And what you can see there is that on a daily cycle, you're getting this wide variation in temperature, in addition to changes in mean temperature. So these mussels, it's important to note, are in tide pools, so they're never in air. These are always submerged mussels. Yet they're undergoing these wide changes in temperature, because when the tide goes down, the tide pool itself heats up as it's exposed to the sun. But all my experiments are done in water, and our focus is on tide pools. So we're interested in thermal change and under aquatic conditions. So what I'm going to run through today are just three little vignettes here. I'm going to start off talking about some of the work we're looking at, the effects of stable versus fluctuating temperature regimes and their effects on mussel growth. And then I'll add a little extra at the end there, showing some of the patterns of gene expression that go hand in hand with that growth. After that, I'll give a little bit of exploration in how we actually choose to approximate fluctuation in the lab. So there's a big difference between temperature fluctuation in the field and what we can do in the lab. So I'll talk a little bit about some of the work we're doing there to try to shore up our experimental approach there. And then last bit, I'm going to talk not just about spatial or temporal variation in temperature. I'm going to talk as one example of just how important spatial variation is. I'm going to look at tissue-specific differences in temperature within a mussel, and what how that can affect some of the biology. All right, so let's get started with that first point. We're going to talk a little bit about stable versus fluctuating temperatures and their effect on performance in these mussels. So here's a graph of physiological performance of an ectotherm from Ray Huey. So on the x-axis, you have some environmental factor here, it's temperature. On the y-axis, you have some measure of physiological or ecological performance. And so that could be growth, it could be respiration, it could be anything that's sort of related to fitness. And so you get this familiar distribution. There's certainly an optimum in the center of that distribution, but it's not just that mean value that matters. There's a shape to this distribution that's important. So it's not just the optimum, it's the breadth of that optimal peak. It is going further to the right and left, the upper and the critical temperatures that the organism can tolerate, and so that defines its physiological tolerance range. And so this is an important type of graph. It's one that you can It's important both experimentally and theoretically. You can construct a graph like that through experiments done in the lab, looking at performance under different temperatures, for instance, and that is very useful. But the graph is built on conditions in the lab that are stable. So when you do this, you're going to measure performance at, say, 5 degrees, and then 10 degrees, and then 15 degrees, and then 20. Each one of those temperatures is stable. And when we do experiments in the lab, we try to keep those conditions controlled and within some parameters, right? And so if you want to say it's 5 degrees, you want to have a nice stable temperature. That's fine, and that helps us create this graph, but it calls into question about that assumption. Is that the best way to be measuring these performance graphs? And so this is a performance graph for a curve for stable temperatures. What would that curve look like for fluctuating temperatures is the question. And so you can do that experimentally. It would be nice if you could use that stable curve to try to make predictions about what the fluctuating curve would be. So we did both. The first thing we did is that we did a little experiment where we tried to look at growth of these mussels under stable and fluctuating conditions. So these were mussels that were collected in Rhode Island. We brought them into the lab, we grew them up for 50 days and measured their growth. So these were just little tiny mussels about 1 centimeter in length. We ran them under a series of stable temperatures, just like we normally would do to make a curve. And then we ran them under a few fluctuating temperatures to try to see if we could get some picture into what that curve might look like. Fluctuating temperatures, we change the temperature every 6 hours. It was plus or minus 5 degrees from each of those numbers. So if it was 10 degrees, it was between 5 and 15. If it was 15, it was between 10 and 20. So plus or minus 5 degrees, and we changed those temperatures every 5 or 6 hours between the min and the max. We had 3 replicates for each treatment. We had 20 mussels per replicate, and fed them every day. So that experiment came out like this. So this is looks familiar to that performance curve that I just showed you on the first graph. Got temperature on the x-axis, growth rate on the y-axis there. So we've got a nice curve that shows a peak and then a little bit of a downturn. The numbers there indicate survivorship over the experiment. And so at the lower temperatures, we didn't have any mortality. All those mussels made it through the experiment. But as we started to get up towards 25 and 26 degrees, those are those last 2 points, we started to see some mortality. So that is consistent to what we see, at least in our lab. 26 degrees is pretty much the threshold. They don't survive past that temperature for us very often. Just did a simple fit of that data, and looks pretty nice. So the question is now, what about the fluctuating curve? What would that fluctuating curve look like? Well, trying to just take the average for the stable curve has its challenges. So this is based on this concept called Jensen's inequality, where if you just look at the average temperature between 2 temperatures on that curve, it might actually be lower or higher than the temperature between those two sides. So the idea is that the function of the averages is not equal to the average of the 2 functions. So you can get, based on whether that curve is facing upwards or downwards, you can get an overestimate or an underestimate of the true temperature. So fluctuating temperatures might lead to higher growth on the upswing on the low part of the performance curve, but then they might actually be lower than stable temperatures under the top optimum part of that graph that's more concave. So what we did here is we took, there's a couple ways you could try to estimate that. What we did is we took that stable temperature curve we just brute force this, where we picked a ton of points on the temperature range. And so we assumed a fluctuation of plus or minus 5 degrees for this one. And we use that equation there. We use the temperatures to make predictions about what the performance would be at that temperature. Then we averaged all those to get to our fluctuating number. So the orange is our fluctuating prediction, and this is our first crack at it. It broadly shows, I think, what we might expect, which is under low temperatures, you might expect to have slightly higher growth rates under fluctuating conditions compared to stable. But then under higher temperatures, you start to see fluctuating conditions might be a little bit lower than stable conditions. What happens at the very high regions, it's a little hard to say. Like I said, our organisms start to die off pretty dramatically at the top end. So we still have a little work to do there. I mean, it looks like it might be starting to converge with the stable curve, but really we need to do more work on that. So then we add in our fluctuating empirical data. And so here are the averages of organisms or of treatments under fluctuating conditions. So this is plus or minus 5 degrees. And so we certainly see that under low conditions, low temperatures, that the growth rates are probably very similar to what the stable performance is. Once we get to that optimum, though, it does seem like we're not reaching as high a peak at the 20 degrees temperature. So that does seem to be sort of in line with our prediction. And then up to 25 and 26 degrees, we get similar drops, and they seem to be a little bit lower than what they are for the stable. So there does seem to be some difference there between the stable and the fluctuating conditions. That's the first point. The growth is certainly lower under fluctuating conditions than it is under these stable conditions, under these warm temperatures. And that's in line with the predictions that we made about fluctuating conditions. So we're going to certainly continue with this, but as a first approach, at least, the differences between the 5 and the 10 degrees range, and then the peak at the 20 to 25 degrees range, at least look like they might be trending in the right directions, where the fluctuating temperatures do look like they're a little lower at 20 degrees and the same or perhaps higher at the 10 to 15 degrees. So yeah, so that's interesting point there, looking at fluctuating versus stable temperature regimes. Now, when we did that experiment, that's in the lab, we didn't have a lot of field data to go with, and so of what the temperatures they would be facing in these tide pools would be. So while we were doing that experiment, we were monitoring temperatures in the tide pools. And so here's that site at Rhode Island. You can see that tide pool there has some mussels in it, and at high tide, the water comes in and covers that tide pool up. But when the tide goes out, it's high and dry. And so when it's high and dry like this, it's sitting in a rocky substrate, and it's a sunny day, that tide pool can heat up really fast. It's kind of like a brick oven. It just gets extremely warm, extremely fast. So what I've got here, the first graph on the top of the panel is looking at this tide pool. How much time is it spending submersed by the tide, the open ocean, and how much time is it by itself above the tide level, exposed to the air? And so even though the mussels themselves are always underwater, the tide pool itself is going through periods of heating and cooling every single day. So the red is indicating periods when it's in the air and heating, and the blue are periods where it's submerged and probably pretty cool and well mixed. So that's happening every single day. What the consequence of that is in graph number 2. The temperatures in that tide pool are changing. And what you can see here, this is a close-up of a couple months worth of data, and you can see that the temperatures are changing every single day. And that's a really, really wide range of temperatures. not what we would have expected at all. Some of those temperatures are going up to 35 degrees or higher. If you ask any mussel biologist if a mussel could survive at 35 degrees, they certainly would say no, right? Some of the work that I showed you in that previous slide showed 25, 26 degrees is where they were kind of dying out. So they're reaching these high temperatures every day, but then they're going back down some 15 degrees cooler when the tide comes in. So really wide temperature range, and yet they seem to be doing fine. When we look at mortality and survivorship over this 2-month period, very few of the mussels died. Only a couple mussels at the small range died for this entire 2 months. Now, maybe the mussels that were weaker would have died before we started this experiment earlier in the summer, but at least for our period, those mussels are undergoing this dramatic fluctuation every single day and doing just fine. And I put the graph at the bottom there as comparison. These are temperatures in the mussel bed themselves. So the mussel bed is down in this picture closer to where the water's edge is. And so even though those mussels are exposed to the air, they are a little bit lower down, and so they don't experience quite as big a range. So these in temperature. So these mussels are experiencing a massive fluctuation of temperature in these mussel or in these tide pools. And yet, when you go to the tide pool and you look at these mussels, they seemingly are doing fine. This is a set of mussels in a tide pool where it's very warm. I think it was around 30 degrees in this one. And those shells are open. So you can see the black shell and you can see the brown mantle, the siphons. They're open. I don't have confirmation if they're physiologically more or less active than normal. We'll have to follow that up. But at least just by first look at it, they're open and their siphons are wide. Usually when they're stressed out, these mussels will close up, clam up, and wait for the conditions to pass. So by all accounts, they seem to be doing fine. So how they're surviving these temperatures is part of what we're trying to get at. So we brought some of these mussels back into the lab and just did a first survivorship experiment. And so the adults under stable or fluctuating conditions, you see that under stable temperatures on the x-axis, the survivorship for adults under stable temperatures were pretty good, 100% all the way up to about 25 degrees, like I said, and then 25, 26 seems to be the threshold for these mussels, and they can't survive any longer. All right, and then the fluctuating conditions, we see that the mussels seem to be doing a little bit better under that 1 point there, 27.5 degrees. All right, sorry, I just see a question. These are both regions combined. Any differences between populations? Yeah, so there are differences between the two populations. I think you're talking East and West Coast here. So I'm answering one of the chat questions here. Yeah, so the temperatures out west at our Friday Harbor labs are typically colder than they are in the Northeast. So the Northeast, they're experiencing higher temperatures. So the thermal responses are displaced a little bit higher and lower, with lower being on the West Coast and higher being on the East Coast. The fluctuation, the differences in fluctuation between the two sites, I am still trying to get a handle on that. So I don't know that there are differences to that degree. But just on a very broad sense, yes, the ones that I was pulling out of Rhode Island have a little bit of a higher average that they're used to. Okay, sorry, that was a little awkward, but thanks for the question. Okay, and then juvenile mussels under these temperatures, they seem to die out a little bit at lower temperatures. And so instead of the 25 degrees, they seem to somewhere between that 50 to 20 degree be being lost. And so that didn't seem to have as much advantage under the fluctuating conditions. And so we don't know the mechanism behind that just yet. That's something we're going to follow up with is developmental stage adults versus juveniles. There seems to be a little bit something different going on. We don't see a ton of juveniles in these tide pools, but we need to monitor them through the season to see what's going on for real. So these mussels are in these tide pools, and they are certainly not dying off. They're not dying off at a high rate, but are they stressed? And so we took some of these mussels and we tried to explore the idea of these fluctuating temperatures on their HSP expression as a proxy for stress. And so we ran these mussels under, again, stable temperatures. In this case, it was 10, 19, and 28. At this early point, we weren't sure what the critical temperature at the high end was, so we went a little bit above what we probably should have. And then we had fluctuating temperatures between 10 and 28, and those temperatures changed every 6 hours. And so we put 3 mussels in each treatment, and we ran this for several days, and we measured patterns of gene expression to see if they were maybe not dying, but if they were actually stressed at all. Were these mussels in these fluctuating conditions stressed? And so here's the results of that. We looked at these 3 genes. HSP70 is the important one. That's the indicator of thermal stress response. We also looked at NADH and APC7, which we're hoping might give some insight into respiration and cell division. So the way this worked, we had 3 stable temperature treatments and 2 fluctuating treatments here, and a fluctuating treatment here. This 3 stable temperatures, 10, 19, and 28, if you look at them, they're run over 5 days, and the expression level of the HSP, let's start with that. So the gene was at 10 degrees, it was normalized to day 1, but over the 5 days of the experiment, it didn't really go up or down that much, indicated by the blue blocks. In the 19 degrees temperature, which is a little warm, the HSP started pretty mild, but then started to increase over time until by the end of 5 days, it had really started to express at higher rates. The 28 degrees, not surprisingly, the mussels were freaked out as soon as we put them in 28 degrees. We got an immediate increase in HSP expression, which suggests they were really stressed out, and then they died after 3 days. So that expression did not seem to protect them much, but did indicate that they were very stressed. The interesting part is this fluctuating panel at the end here. So going from 10 to 28 degrees, the HSP starts off a little bit elevated, but as time goes on, the expression of HSP started to rise more and more. And that was interesting because a lot of times when we have stressful, stable temperatures, we'll see an increase in HSP expression initially, and then it will just start to decrease over time. They stop producing it if they are held in bad conditions too long. This one, though, it started to increase, and it just kept increasing all the way to the end. So they seem to be doing fine in those fluctuating conditions, but they do seem to be pretty stressed out in those conditions. So certainly there's something different going on between the stable temperature conditions and those fluctuating conditions. There's differences in growth. There's not much change in mortality, but there's certainly some differences that if we peel down into the level of gene expression, there does seem to be some stress going on. So we're following up with this, looking at a little bit at feeding rates and respiration rates. We're going to try to make an energy budget model to really see what the dynamics are under stable versus fluctuating conditions. But this is giving us a little hint, I guess, of what mussels are experiencing in these conditions. And I wanted to think about how general these conditions might be, and this sort of gets a little bit to the question in the chat. So these are temperatures, high and low temperatures for any 24-hour period in data taken from tide pools from different parts of the coasts, East Coast, West Coast. And so these are all tide pools that have mussels in them. So this is a very specific type of niche graph. These are only tide pools where we have confirmation that there are mussels in there. And we look at the temperature data, and we look for the day where it has the widest range. And so this is the widest range that we find at these places. But what it shows is that that a lot of these mussels, no matter where they're living, do experience pretty wide variation in temperature in these tide pools. I mean, eventually, once you go to the lower latitudes, it starts to shorten up that range. But certainly, for a lot of the temperate sites, whether you're on the East Coast or the West Coast, we do see these wide ranges in temperature. And like you can probably see a little bit, there's a little difference there in Narragansett versus Friday Harbor, and that displacement I was talking about in temperature versus the East and the West Coast populations. So it's a little bit different. So a lot of this temperature is from Brian Helmuth's group at Northeastern and his colleagues, but this has helped paint a little bit of picture that this variation in tide pools seems to be fairly common stuff. So that was just a first whack at stable versus fluctuating temperatures. There are certainly different biological responses to those different thermal regimes. What I want to talk about next is how we actually are exploring how we approximate thermal fluctuation in the lab, because this is a big issue that we're having. Be curious to see if anyone's got any suggestions for this. So here's some temperature data from a previous experiment of mine. And I was looking at stable versus fluctuating conditions. And so we had 3 stable temperature conditions, 15, 20, 25 degrees. And then we had fluctuating temperature conditions with a mean of 20 plus or minus 5 degrees. And you can see that's a really blocky temperature form, right? It goes up and it goes down. I can play around with the amplitude, I can play around with the frequency, but it's basically going between some minimum temperature and some maximum temperature. And especially for that blue one, the mussels are stuck either at low temperature or at high temperature. And so how appropriate that is kind of, I think, what we're calling into question here a little bit. You know, you can play around and you can make a sine wave to try to soften that blockiness from it. But you're still going between these minimum and maximum temperatures. So the question is that appropriate? Is there a way that we can improve that? So I've showed you that we've taken a lot of temperature monitor from the tide pools. And so what we've done is we've taken those temperature logs and we've built a little flow chamber that can respond fast enough to change temperatures to approximate what we see in the field. So we've got a little flow chamber that can change temperatures every 5 minutes. And that seems to be fast enough to approximate what we see in the field. So what we've got here on the top graph here are different types of temperature treatments we could have. The field data is the data that we've taken off our temperature loggers. And so you can see that it's this kind of asymmetric rise and then fall, right? It's not just your simple Gaussian. Then you've got the block idea, which is typically what I use, which is that minimum to maximum form. And then the last one is a sine wave, and to try to make it a little bit more curved. I'm going to talk about comparing the field data to the sine wave data. So what we did is we took our mussels and we put them in field temperature regimes or this block regime. And then we measured rates of respiration to see if there were differences between just the type of fluctuation. So not stable versus fluctuating. Now we're looking at the different types of fluctuation. Does that matter? So we took just 2 days, one from April, one from July, from our field temperature data, and we programmed that into our flow chamber. We made sure that the block and field data have the same mean, but the shape is certainly much different. We ran a 12-hour trial. We measured respiration rates over that 12 hours by a single muscle. And here's what we got. This is kind of interesting. So what we've got are the lab temperatures here. The dark blue are field temperatures, the light blue are block temperatures. And the respiration rates are measured on the y-axis. So like I said, we had a spring temperature, which was April, and a July day temperature, which was warmer. And the field temperatures in April gave a lower respiration rate than the block temperatures. That was accentuated in July, when we certainly had higher temperatures just in general, and there was a bigger difference between the field temperature regime versus the block temperature regime. And so part of that could be that there's an abrupt change with those blocks, and so you're shocking the organism physiologically, but also that block temperature regime is going between a minimum and a maximum. And I think that maximum is probably a bit of a problem area, and that you're holding the organism at its maximum temperature and it's getting stressed out. And even if you pull it back down to the minimum, you're probably getting an overestimate of respiration rate. So this is our first data on this. We're going to continue looking at the differences in these different types of thermal regimes, but the early results are showing that it looks like field temperatures might be having a different effect on the physiology of the organisms, and that it depends on the time of year. Our April and our July temperature regimes yielded different results. So we're going to continue looking at that. All right. So we talked a little bit about stable and fluctuating conditions, and then a little add-on to that was the type of thermal fluctuations in the lab there. So I'm just gonna check the chat thing question here to see if there are any questions, if anyone's got any stuff on that before I launch into the last vignette.
I think we're all good to keep going,
Good.
Mike. Thank you.
Right. Okay. So the last part I want to talk about was the effects of warming on variation within the muscle. So I gave you a bunch of field temperatures measured in these tide pools, and we measured them with loggers. There's a picture here on the left that my colleague Brian Helmuth was using to measure muscle temperatures in the bed. You can see it with the whiteout number on there. And so we're often looking to try to predict the temperature of the muscle, right? We've got environmental temperatures, but what does that mean for the muscle itself? And so we're out there measuring individual muscles, trying to see what their temperatures are. As we're doing that, though, one of the problems that you come across is there any variation within the muscle itself? And as it turns out that that is actually true. When we're looking at temperatures and their effects on these muscles and how it stresses them out, the temperatures of the different tissues, in fact, are potentially different. So here's a muscle that I've cut open, and you can see the different tissues. We've got the mantle tissue lining the lip of the shell, and that's where it makes its siphons. Gill is that dark yellow material, important down both sides of the shell. And then the foot is that dark material. And on the right-hand side there, you can see the foot gets extended, helps the muscle actually move around its environment, make small adjustments if it needs to, and is really important in laying down its byssal threads. So byssal threads are what hold it onto the rocks. So the foot is very important to a lot of the biology of the muscle. So the question is, We've talked a lot about temporal variation in the thermal response of the muscles. It's other spatial differences as well. And so the students measured the temperatures of different tissues in the lab after we had heated up the muscles in air in this case. So this is aerial temperature. So they're under a heat lamp basically to approximate what they would be like in the air. What you see is that the air bars are certainly overlapping here, but there does seem to be a little bit of difference between the mantle versus the gill on the foot. We only have a few samples for this graph, and my students are continuing to try to fill this one in. But hopefully, if these patterns hold up, we do see differences within the muscle itself as it is exposed at low tide to these different temperatures. And connected with those different temperatures, we then took samples from each of those different tissues and measured HSP expression. And we did it at 2 time points, 20 minutes after they've been warmed and 50 minutes after they've been warmed. And at 20 minutes, HSP expression relative to just an unstressed muscle was pretty low on the foot, but had been elevated in the gill and the mantle, which was interesting. The mantle was quite a bit warmer, so that's expected, but the gill also showed some increase in HSP expression. After 50 minutes, though, it was a different story. Basically, in all 3 tissues, they had shut it down, and so HSP expression had really ceased under these conditions. Now, these are pretty extreme heating conditions that we exposed them to. We tried to heat them up as fast as we could. So we'll have to try some range of temperatures, but it does seem like depending on the time and the tissue, we can get a different snapshot into how these muscles are being stressed out or not. So that's something that we're gonna have to start taking an account of as we monitor these muscles. So those are the 3 stories I've got. 2 stories about temporal variation, 1 about spatial variation. The muscles certainly are exhibiting distinct responses to fluctuating temperatures versus shifts in mean temperature. It seems like those fluctuating temperatures are affording periods of relief and recovery for those muscles to not undergo so much mortality, but that's something that we're still working out to get the mechanisms underpinning that pattern. And all this work just suggests that these fluctuating conditions should be included in the experiments and models we do that are seeking to say something about thermal stress in these intertidal organisms. So that's it with that. If you have any questions, I'm happy to take them now.
Thank you so much, Mike. It was a great talk about something that I feel like we don't hear a lot about from University of Washington, or those of us who work on groundfish. So
really dominant.
Yeah, we can go ahead and take questions in the chat if anyone is interested, or you can raise your hand and we can unmute you. I can go ahead and get us started and selfishly ask you a question first, Mike. From your plot that showed differences in respiration when in April and in July, how you had that greater difference at warmer temperatures between was it the field-based temperatures and then your block?
The block ones were elevated. Yeah, they're shocking them, I think, a little bit.
Yeah, so I guess I'm curious, with the slightly warmer temperatures experienced by the mussel populations on the East Coast, which you might think might be in a better situation with global climate change and warming temperatures.
Yeah, so that's the question. So the question, are the ones on the East Coast, if they're slightly warmer temperatures, are they more adapted to those, or are they closer to the limits, right? And so to get to that, what you really need is you need those performance curves for each species, which kind of gets back to the question in the chat, because you need to see where's that optimum and where's that drop off, right, in their performance. And then where do they sit normally in the environment? So the question, you really need another performance curve from the West Coast mussels compared to the East Coast mussels to answer that question, I think, is the point there. I don't know which ones are closer to their maximum right now.
Okay, that helps. Thank you. All right, any other questions from the audience? Again, you can put them in the chat or raise your hand. You can just wait a second, give people a moment. I was curious, you focused on temperature, but I'm trying to remember dig deep to what I know about mussels. I was curious about some of the other variables that you might be interested in taking a look at.
Question. Yeah, so temperature is just a nice easy one. It was the one for the undergrads that we saw, I think, most readily. But you're right. I give a little nod to the pH is changing in those tide pools every day, right? When we think about ocean acidification, we often hear from the oceanographers who tell us how much the ocean is changing sort of in the open ocean, right, with pH. But in these tide pools, they're changing a ton every day because there's photosynthesis and there's respiration going on in those tide pools every single day. So night versus day, they're going through a big change in CO2. And so pH is definitely one of them, and then oxygen is another. And so those two are things that we're looking at along with temperature. Working at Friday Harbor with Emily Carrington and another collaborator, looking at mussel beds. And as mussel beds slow the water and are doing their normal physiology, are they changing the conditions under them? And pH, oxygen, and temperature, are they creating these sort of bad microclimates underneath them, which is a different play than this tide pool idea. But the reason we think that's important is that Emily's got some data showing that the byssal threads can weaken with the pH, the oxygen, and temperature. So we're seeing if there's a biological sort of loop here where they're causing their own demise a little bit under climate change.
Oh, that's really interesting. Awesome. Well, I don't know that we have any other questions. I just wanted to thank you again for coming to.
Thank you very much.
All right. Have a good one. Thank you so much. Thanks everyone for coming to today's seminar. Bye.